next speaker is Paul Hebert. Paul is the director of the Biodiversity Institute of Ontario, which we've already heard about. And Paul will talk about barcoding and... Thank you, Mark.
Glad to see I have a PowerPoint slide on the screen as opposed to... I just want to spend 10 minutes talking to you about progress on the barcoding front for members of the animal kingdom, the most speciose, or so we think, of the divisions of eukaryotic life on the planet. The happy news is we have a target region that we selected a few years ago, and looking back at results since then, it seems that it's performing in a reasonably effective fashion. In fact, if I'd been asked 3 years ago if things would be as simple as they've turned out to be, I would have said almost certainly not. I expected much greater complications linked to shifts in rates of evolution and among different taxonomic groups and perhaps different biologies of organisms. I've been startled at how simple things seem to be. So we've been focusing on the insights you can gain from this 648 base pair region at the 5' end of the cytochrome C oxidase 1 gene. As has been mentioned, we have been supported by a number of agencies here in Canada and outside. That's allowed us to create an analytical platform that's been pushing hard, not in big genomics context. I'm afraid we don't rival the veterinarian sort of platforms, but in terms of biodiversity platforms, we've done, I think, a substantial amount of work, and the Barcode of Life database last week reported about 260,000 specimens having been analyzed at that point in time, and about 29,000 species registered. Now that's a tiny fraction of eukaryotic life, and of course that's why we're here today, I think, to and over the next couple of days, to imagine how we might drive up these records from a few 100,000 to many millions, and to drive up species coverage. Now, animals is where it began, and if one takes a look at the taxonomy browser, there are 261,000 records. There's a huge chain of records to the animal kingdom from large segments of the planet. There are still some areas that need deeper coverage, obviously. The plants, fungi, and protists are just gaining activation, and we're going to hear more about them. So most of the insights we have today derive from animals. So what have we learned? I'm going to spend just a couple more minutes talking about a few of the issues that we've been investigating. One is the idea driven initially by trying to develop an efficient mechanism for species identification. So I'll talk a little bit about that, and I'm going to argue in the course of that, in those few slides, that DNA barcoding is working surprisingly well in terms of delineating species, those recognized by prior taxonomic endeavors, and revealing species that may have been overlooked by prior work. Why is that? And I'm going to argue that the real success of barcoding relies upon this strident difference between the amount of sequence variation between species and within species. It wouldn't have been expected that there would be such a deep chasm. And then finally, we all know that most life on the planet is not yet registered, 1.2 million out of perhaps 10 million species. How can we use the insights that we gain in the course of using these ground truthing studies on really well taxonomically understood groups to probe unknown life? So those are the 4 points I'd like to touch on. Here's a sort of one of our earlier studies done with Mark Stokel and colleagues that looked at North American birds, a temperate assemblage of species, and this sort of pattern of very small amounts of variation within members of a species. These specimens on average were 1,100 kilometers apart, and these deep differences between members. These are all members of the family Scolopacidae, the sandpipers and their kin. And this difference, 95% across now North American birds. We've nearly finished the North American bird fauna. About 95% species level resolution. It's not a perfect approach, but this difference between the within species levels of variation, a fraction of a percent, and the deep divergences even between congeneric species. So that was birds. We began in the temperate zone, and there was some early concerns that these results might not be extendable into the tropics. And happily, we connected with Dan Jansen and Winnie Howicks and moved into Costa Rica, and Dan's long-standing passion and intense study in this area. And one of our earlier works was on Saturnids, but Meredith Hebert here at the University of Guelph got involved and has been very successful in leading a big charge on Costa Rican Lepidoptera. A recent paper looked at nearly 550 species of these organisms in the tropics and the same sort of pattern of shallow variation in individuals and this big divergence, 98% resolution, and this big divergence between congeneric and within species. So the approach appears to work in the tropics as well as the temperate zone. Would it also work in the marine environment? We moved with Bob Ward and colleagues at CSIRO Australia to ask the question about Australian fishes, and the resolution was embarrassingly good, 100%, and this big difference, and the same sort of pattern. So we've now got 260 or 1,000 records or so that look like this, and the pattern is invariant across taxonomic groups, I would say, with very few exceptions. So species identification is working well. Why? Deep divergence is the rule between members of a genus. Species that are often quite difficult to tell apart morphologically are almost invariably showing deep divergence. Why is that? The fossil record showed us that species are typically have lifespans of millions of years. That's all we're rediscovering. Something that was well known, that species are old. So I don't think that's part of the surprise of barcoding. We're just gaining an insight into the fact that species aren't bubbling up overnight. They've been fellow travelers on the planet for millions of years. The surprise has come, I think, on the other end of the equation. It's the lack of variation within species. We knew our own species showed remarkably low variation, the so-called Eve hypothesis. But this result, this lack of variation within species, was certainly not expected to be a general phenomenon, but it is. Almost every species we have looked at shows fractional percents of sequence divergence. In the case of our own species, it was linked to population collapse. That seems increasingly unlikely as one observes this phenomena across all life. I would say there's a growing sense that this lack of variation may be due to selective sweeps, the stripping out of variation on a recurrent basis in animal populations.